We're going to talk about the process of semi-conservative DNA replication. Whenever a cell undergoes cell division, prior to that cell division, the DNA must be faithfully copied and 2 identical copies produced. This will happen in all living cells, whether it's a simple bacterium that just has 1 circular chromosome, or whether it's a more complex cell, a eukaryotic cell that has multiple linear chromosomes. So before we look at the process of semi-conservative replication of DNA, let's just refresh very quickly the structure of a DNA molecule. DNA is a polymer built from nucleotide subunits, and each nucleotide is comprised of a phosphate, a sugar and a base. So let's just draw a simple chain of nucleotides joined together. So phosphate, sugar and base. So that's 1 nucleotide. So there we have 1 nucleotide. So when a strand of DNA is synthesized, nucleotides bond together, forming the phosphate sugar backbone with the bases in the center of the double helix. So this is just one side of the double helix. And in a minute I'm going to finish that off quickly. So there we have a simple strand of double helix, 2 strands of DNA that are complementary base pairing. We can assign the bases, so this one might be A bonded to a T, a G bonded to a C bonded to a G, and a T bonded to an A, for example. It's a sequence of bases in DNA that are important in coding for specific proteins. You should be able to describe the structure of DNA as a double helix comprised of 2 strands that complementary base pair. You should also be able to describe the fact that these strands are held together by relatively weak hydrogen bonds, and it's the hydrogen bonds between those strands that can be broken in order to expose those bases, and that's what we're going to talk about next in DNA replication. Let's look now at the process of semi-conservative DNA replication. I'm just going to draw this double helix molecule more schematically. So we know that DNA, the structure of DNA is a double helix with the sugar phosphate backbone and the base pairs in the center of that molecule. The base pairs, or the bases, being complementary to each other, A bonding to T, G bonding to C. Now, in order for DNA to be copied, to make a faithful copy of itself, the first thing that needs to occur is that that double helix needs to be unwound, untwisted and unzipped. And the way that cells do that is to use an enzyme called DNA helicase. Whenever you hear the ending A's, you should be thinking of an enzyme, which is of course a biological catalyst that speeds up a chemical reaction inside a living thing. So DNA helicase will come along and unwind this double helix. So this is part of the DNA that's not yet unwound. This will happen bit by bit as synthesis is occurring. And so when the DNA is unzipped and those hydrogen bonds are broken, this leads to the bases along those strands of DNA being exposed. And that's important because the next enzyme, known as DNA polymerase being an enzyme that catalyzes the synthesis of a polymer, in this case a polymer of DNA. So DNA polymerase will come along, and now that the bases have been exposed, DNA polymerase will start to bring in, I should just draw some free nucleotides. So there are free nucleotides. If we're talking about a eukaryotic cell, they are located in the nucleus. And so DNA polymerase will bring those nucleotides in and use complementary base pairing, again A to T, G to C, to start synthesizing a new strand using that existing DNA strand as a template, and therefore produce a brand new strand of DNA that's complementary base pairing to that template strand. The same thing will happen on the other strand. Does happen a little bit differently. It happens in short little fragments because of the direction of DNA, and then those fragments are joined together. So what will happen if the direction of synthesis is occurring in this direction? So a little bit more of that double helix will unwind and unzip using the enzyme helicase, and polymerase will keep adding on those nucleotides until two complete copies of DNA are produced. It's important to remember that in DNA replication, the whole of the DNA is copied, not just a part of it. And that's because the new cells produced need a full, complete copy of the DNA so that it has the instructions for normal functioning. So what we end up with are two strands of DNA. And it's important for you to understand the concept of semi-conservative and what we mean by that. So two identical double helices, identical to the parent. And the reason we say that DNA replication is semi-conservative is because in each of those two new double helices, or double helix molecules, we have what we call a parental, or sometimes called the old strand. And then we also have the newly synthesized strand built from those free nucleotides. So semi-conservative replication refers to the fact that half of this DNA has been conserved from the original DNA that's been copied, and half of that double-stranded helix has been newly made using these free nucleotides. So hence the term semi-conservative. You might like to practice describing this process in words, some key words to use. You don't necessarily need to know the names of the enzymes, but you need to understand that the DNA double helix is unwound and unzipped to expose the DNA bases. It's important to use the word template because each of those strands, once the bases are exposed, each of those strands will act as templates for DNA polymerase to start synthesizing a new strand. You also need to explain why we call DNA replication semi-conservative, in that it has a conserved parental strand or an old strand together with a new strand, and hence we've produced these two identical molecules that are identical to that original double helix.